This video will be a continuation in our series about the nervous system, and this video will cover synapses in a little bit more detail than they were covered before. So before we took a look at the structure of a synapse in relation to the structure of a neuron. Here we're going to take a closer look at the synapse, and we find that we actually have two major types of synapses: electrical synapses and chemical synapses. In an electrical synapse, our presynaptic membrane is in direct contact with our postsynaptic membrane through gap junctions. This is going to allow our action potential to come down our presynaptic membrane and flow directly through our gap junction to continue the action potential in our postsynaptic membrane without any pause, halt, or need for the conversion of our electrical signal into a chemical signal and then back into an electrical signal. So these are going to have the fastest transmission times because the action potential does not need to stop and wait for that conversion we spoke of earlier. Electrical synapses are not very common. We do see them in some areas in our brain, but they are not widespread throughout our body. The majority of synapses in our body are going to be called chemical synapses. In a chemical synapse, there is a gap between the presynaptic and the postsynaptic cell that we call the synaptic cleft or the synaptic gap. Because we have a gap, we are going to need to send neurotransmitters across that gap so that our presynaptic cell can communicate with our postsynaptic cell. So in our picture, we are going to have an action potential come through our presynaptic membrane. This action potential will cause synaptic vesicles filled with neurotransmitter to fuse with the presynaptic membrane, releasing neurotransmitter into the synaptic gap. That neurotransmitter will bind onto receptors on the postsynaptic membrane to cause an action potential in our postsynaptic membrane. So our chemical synapses are reliant on neurotransmitters to carry our message from our presynaptic cell to our postsynaptic cell. These chemical synapses can appear in many different places, and our presynaptic neuron can synapse with a dendrite, a soma, or an axon to form an axodendritic, an axosomatic, or an axoaxonic synapse. So this tells us where our synapse is occurring. An axodendritic synapse is going to be a synapse between an axon and a dendrite. An axosomatic synapse is going to occur between an axon and a soma. And finally, an axoaxonic synapse is going to occur between an axon and an axon. Not only can we classify our synapses by where they occur, we can classify them by the neurotransmitter used. I use the abbreviation NT for neurotransmitter. Here in our picture, we have a cholinergic synapse. What this means is that our presynaptic cell is going to release acetylcholine as our neurotransmitter, and our postsynaptic cell has cholinergic receptors. That is, receptors that bind to acetylcholine choline. So we see the word choline in both our neurotransmitter and our receptors. So cholinergic receptors bind to acetylcholine. We also have adrenergic receptors. Adrenergic receptors are going to bind to adrenaline or noradrenaline. Another word for adrenaline is epinephrine, and noradrenaline is also called norepinephrine. So then our postsynaptic cell has adrenergic receptors. They're called adrenergic because they bind to adrenaline. Those are the two main types of chemical synapses that we will cover in anatomy and physiology. But what happens to our presynaptic neuron and our postsynaptic neuron after that action potential has passed? So our action potential is going to come down our axon and cause the release of neurotransmitters which stimulate our postsynaptic cell and cause an action potential in our postsynaptic cell. What next? You actually have to turn off stimulation, or our postsynaptic cell will continue to fire action potentials indefinitely, and you don't want that. So what happens is our neurotransmitter is going to bind onto our postsynaptic cell, but it only binds for about a millisecond, and then it dissociates and detaches from our receptor. So why doesn't that neurotransmitter then just immediately bind onto another receptor? Sometimes it does, but we don't want that to continue forever. And so we have a couple of different mechanisms to remove neurotransmitters from the synaptic gap. One way is through diffusion. In diffusion, our neurotransmitter simply leaves the synaptic gap and joins the interstitial fluid at large. These neurotransmitters are absorbed by astrocytes in the central nervous system. What we see going on in our picture is called the reuptake of neurotransmitter. So either our pre or post synaptic knob is going to take our neurotransmitter back inside and put them back into secretory vesicles. This can happen through a protein. So here we have a reuptake transporter, or it can occur through endocytosis. A third way that we can remove neurotransmitter from our synaptic gap is to break down that neurotransmitter in the synaptic gap. So here we have an enzyme called acetylcholine esterase. Acetylcholine esterase is going to take acetylcholine and break it into its component parts of choline and acetate. Those parts are then reabsorbed into either our presynaptic cell or into our postsynaptic cell. So we've broken down our neurotransmitter so that it can no longer stimulate our postsynaptic cell, but we are going to reuse the parts. If you have any questions regarding any of this material, please do not hesitate to contact your instructor.